Okay, so in this video, we're going to look at Punnett squares involving multiple allele inheritance. So these are traits that have more than two possible alleles. So instead of having one dominant and one recessive, they'll have more than those two. So maybe two dominants, or a dominant and two recessives, or a dominant and four recessives. So we'll take a look at some examples of Punnett squares involving multiple alleles. So for these examples, we're going to use blood type for all of the practice problems. And blood type's a little bit unique because we have three alleles. We have allele A, allele B, and allele O. Now, if you might remember from our notes and our video presentation there, that A and B are dominant, so therefore they're co-dominant, and then O would be recessive. So we have three possible alleles. Remember that A and B are co-dominant and O is recessive. When we do Punnett squares with blood type, we don't actually use A, B, and O. So you might see a few examples of Punnett squares where the letters A, B, and O are used, but we actually use a letter I to represent a capital letter I to represent dominant and a lowercase I to represent recessive. And since we have two dominants, we have to have two kind of superscripts that looks like an exponent. So for an individual who has blood type A, their genotypes could be IA, or IA little I. Remember that A is dominant, so we have a dominant or capital letter I, and then we have the superscript A to represent type A. We will have a superscript B to represent type B. So individuals with B blood would be homozygous IB or heterozygous IB little I. So the dominant will mask the recessive and we'll have the B type blood. Individuals who are AB will have IA, IB. They need both A and B alleles to express the AB blood type. You might remember that blood type now has both A proteins and B proteins on the surface of the red blood cells. O is universal donor, but it's also recessive. So we have little I, so no dominance present. So essentially our key might look something like this. Okay, so capital I with a superscript A means that blood type has, their blood cells have protein A. Capital I with a superscript B, those blood cells have protein B. Little I has neither of those proteins. There are other proteins on the surface of the cells, just not proteins associated with this portion of blood type. Do keep in mind that blood type does involve the A and B allele, but it also has that positive and negative. You might know that if someone has type A positive blood, for example, that's because there's actually two different genes that contribute to blood type. One of them refers to the ABO proteins. One of them refers to a different protein called the RH protein. And you'll see that in an activity we'll be doing next week. So we'll do part A as the example, and you can do the rest for your practice. So a woman who is homozygous, so that means two of the same alleles, we know that, for blood type B. So remember up top here, we can have a homozygous for B or heterozygous for B. So the only way to be homozygous and have B blood is if you are dominant and have two IB alleles. She marries a man that is heterozygous, okay, for type A. So type A could be IA or IA little I. If he is heterozygous, he must have two different alleles. So he is IA little I. We're going to show our Punnett square and the chances that the child will be type AB blood. So we're going to set up our Punnett square like normal. I have my key up top. I'll just leave it there. Okay, so these look a little different because now we're going to have both the I and the superscript that we need to bring down. So we're going to put our first genotype, IB, and then our second genotype, IA, and then our little I. We'll fill in our Punnett square. We'll keep the A's first. So IA, IB, little I, IA, IB, and little I. So two of our offspring, two of the F1 generation here, are heterozygous co-dominant, so they have AB. And then two of our offspring here, 50% chance of the individual being B, but heterozygous, therefore they can pass down the recessive. So here's our question. What are the chances that the offspring, the child will have type AB blood? Well, here are our 2 of the 4, so 50% chance the child will have AB blood. So I'll do another example with you since these are a little bit tricky, and then I'll leave the rest for you to do on your own. So for letter B, a man with blood type O marries a woman with blood type AB. So the only way to have blood type O, because it's recessive, is to have no dominant alleles. No dominant alleles means our individual here is going to be homozygous recessive, little I, little I. For a woman to be blood type AB, the only way to have blood type AB is to have a dominant A allele and a dominant B allele. Any other genotype will give you a different blood type. We're going to show our Punnett square and then figure out the possible phenotypes of their children. So with our Punnett square, our first parent is homozygous recessive, they are O blood. Our second parent is AB blood, so they are heterozygous codominant. We can fill in our Punnett square like normal. Don't forget to bring along the superscripts if it is dominant, because they matter. So we can see that 2 of our offspring here are heterozygous type A, and 2 of our offspring are heterozygous type B. The question is to tell how many, or what are the possible blood phenotypes. So the phenotype is the express trait that our offspring can have. All right, so the possible phenotypes or blood types of our offspring will be blood type A, which would be, in these 2 examples, they're heterozygous type A, or type B in these 2 examples. All right, so that leaves C, D, and E for you to practice and turn in with your assignment.